Welcome to another chapter, the fundamental unit of life. Let us see the structural organization of the cell. We will begin with the cytoplasm. What is the cytoplasm? The cytoplasm is a jelly-like substance that fills the space between the cell membrane and the nucleus in a cell. It is mainly composed of water molecules, salts, and various organic molecules. Cytoplasm provides a medium in which the cell organelles are suspended. The cytoplasm is all the material within a eukaryotic or a prokaryotic cell. It is enclosed by the cell membrane including the organelles and excluding the nucleus. It is fluid content present inside the plasma membrane. It contains many special organelles, each of which performs specific functions for the cell. Cytoplasm is just like the playground, where all the organelles live, move, and work together. The functions of the cytoplasm are: it serves as the site for many metabolic reactions, maintains the cell shape and structure through the cytoskeleton. Cytoskeleton is a network of tiny protein fibers present in the cytoplasm of a cell that acts like the cell's internal framework or skeleton. The cytoskeleton is the cell support system that gives shape, strength, and organization to the cell and helps it move and transport materials inside. What are the functions of the cytoplasm? It suspends and protects the organelles, facilitates the transport of materials, and stores essential nutrients and chemicals. Here are some interesting facts about the cytoplasm. Cytoplasm is slightly alkaline with a pH of 7.2. In prokaryotes like bacteria, the cytoplasm is where everything happens, since they don't have a nucleus or a membrane-bound organelles. In eukaryotes, the cytoplasm along with the nucleus forms the protoplasm. Thus, the protoplasm is the living content of a cell in the eukaryotes. Now we will discuss the other cell organelles one by one. The endoplasmic reticulum or the ER. The endoplasmic reticulum is a large interconnected network of membranous tubules, vesicles, and flattened sacs cisternae present in the cytoplasm of the eukaryotic cells. The endoplasmic reticulum or the ER is a membranous network. It is made up of single continuous lipid bilayer. This membrane is directly connected to the nuclear envelope, the membrane around the nucleus. What are the cisternae in the ER? They are the flattened sac-like structures stacked one over the another, and they provide a large surface area for the biochemical reactions. The biochemical reactions are nothing but chemical reactions that occur inside living cells to keep an organism alive and functioning. The biochemical reactions are the life support reactions of the cell. They help in growth, energy production, repair, and survival. What are the tubules in the ER? They are branching tube-like structures which spread throughout the cytoplasm. They help in transportation of the materials inside the cell. What are the vesicles? They are the small bubble-like structures that pinch off from the ER membrane, carry proteins, lipids, or enzymes to other parts of the cell like the Golgi bodies. Now there are two types of endoplasmic reticulum based on the surface. First is the rough endoplasmic reticulum and the other is the smooth endoplasmic reticulum. The RER is so called because its membrane is studded or dotted with embedded granules, organelles called the ribosomes, giving it a very rough appearance. The RER has the ribosomes attached to its surface, and this gives the rough appearance under the microscope, and it is found near the nucleus. The RER is interconnected with the nuclear envelope and extends outward, often appearing as a network of flattened sacs or tubules studded with the ribosomes. The RER is often found in close proximity to the Golgi apparatus, and the main function is protein synthesis and transport. How the RER transports the proteins? Let me explain. The RER has ribosome attached to its surface. That's why it looks rough. These ribosomes make proteins. As the ribosome makes the protein, it threads the growing polypeptide chain directly into the lumen inside the RER through special protein channels called the translocons. In the RER lumen, the proteins undergo modifications. Once the proteins are ready, the RER buds off the small vesicles or the tiny sacs containing the proteins. These vesicles pinch off from the RER membrane and move towards the Golgi apparatus. The Golgi apparatus further modifies, sorts, and packages them for final destinations inside or outside the cell. In simple terms, the RER is like a protein factory plus a quality control room. It makes the proteins, folds and checks them, and then ships them in little vesicles or the delivery trucks for the Golgi body. The smooth endoplasmic reticulum. It is made up of a network of fine tubular and interconnected structures. Unlike the rough ER, it does not have ribosomes attached to its surface, which makes it look smooth under the microscope. The SER usually appears as a meshwork of tiny tubules and vesicles spread throughout the cytoplasm. Its tubular shape allows flexibility and helps it form an extensive network for lipid synthesis, detoxification, and storage of ions like calcium in the muscle cells. The liver cells contain large amounts of SER to support vital detoxification role, which is crucial for maintaining the overall health. The Golgi apparatus was discovered by Camillo Golgi in the year 1898. The Golgi apparatus is an organelle in the eukaryotic cells that modifies, sorts, and packages the proteins and lipids for secretion or transport to other organelles. The Golgi apparatus, also called the Golgi body or the Golgi complex, is an important cell organelle often called the post office of the cell because it sorts, modifies, and packages and transports the proteins and the lipids. The structure of the Golgi apparatus contains a stack of flattened membrane-bound sacs called cisternae. It has two faces. The cis phase faces the endoplasmic reticulum and receives the proteins and lipids from it. The other phase is the trans phase or the maturing phase, which faces the plasma membrane and releases processed materials into the vesicles. It is surrounded by vesicles that transports the molecules in and out. The functions of the Golgi apparatus are protein modification. It modifies the proteins from the rough ER. Lipid transport and processing, processes and sends the lipids to various cell location. Packaging and secretion, packages proteins and lipids into secretory vesicles for transport outside the cell. Lysosome formation, it produces lysosomes, which are the digestive organelles filled with hydrolytic enzymes. The synthesis of complex carbohydrate. In plants, it synthesizes complex carbohydrate for the cell wall. It synthesizes complex polysaccharides, including the components for the plant cell wall like the pectin and hemicellulose. In animals, it is involved in the formation of certain extracellular matrix components. The extracellular matrix is a complex network of proteins and other molecules secreted by the cell that surrounds and supports them, providing structural scaffolding, facilitating cell adhesion, communication and movement, and regulating tissue development, growth, and repair of the tissue. It is a network which contains collagen and glycoproteins, etc. How the Golgi apparatus works. Think of it as a factory shipping department. Raw materials, the protein lipid arrive from the ER. They get processed, they get labeled, modified, and then packed into boxes and then delivered. Now comes the lysosomes. Lysosomes are membrane-bound organelles containing hydrolytic enzymes that act as the cell's digestive system, breaking down waste, cellular debris, and foreign invaders also. The lysosomes were first reported by Christian de Duve in the year 1955. Lysosomes are classified into four main types based on their morphology and function. Primary lysosomes, which are small newly found vesicles from the Golgi apparatus containing inactive enzymes. Secondary lysosomes are formed when a primary lysosome fuses with food vacuole or vesicle to begin the digestion process. The residual bodies or residual lysosomes, which contain undigested material, and the autophagic vacuoles or autolysosomes are formed when primary lysosome surrounds and digests worn out or damaged cellular components. The lysosomes are involved in the digestion of microorganisms by phagocytosis. In the process of phagocytosis, they fuse with a membrane-bound sac called the phagosome, which contains the engulfed particle. This phagolysosome enzyme then breaks down and digests the foreign material such as bacteria, cellular debris within the cell. In certain conditions, the lysosomes start to digest the dead cell organelles. That is why they are called the digestive bags of the cell. They are called the suicide bags because they contain potent digestive enzymes that, if released, can digest and break down the cell's own content. This often happens when a cell becomes damaged, aged, or infected, leading to release of these enzymes and subsequent self-destruction of the cell. Now comes the mitochondria, which is called the powerhouse of the cell. The mitochondria typically has a sausage-shaped or cylindrical shape. They are dynamic organelles that can change the shape, fuse and separate in a process called the mitochondrial dynamic, and can appear as a rod-shaped, filamentous or even thread-like structure. Each mitochondria has an outer membrane, an inner membrane that folds inward to form cristae, finger-like projections, which increases the surface area for energy production, a central fluid-filled space called the matrix. The mitochondria are semi-autonomous bodies because they possess their own DNA, ribosomes, and machinery for protein synthesis, allowing them to replicate and produce some proteins independently also. They are not fully autonomous. The mitochondria uses aerobic respiration to generate the ATP, which is the energy currency that powers the cellular activities. Some cells in some multicellular organisms lack mitochondria, for example, mature mammalian RBCs. What would happen if there was no mitochondria at all? Ever thought? If a cell or organism completely lacked mitochondria, it would not be able to produce enough energy through aerobic respiration, leading to a lack of ATP, that is the cell's powerhouse, and consequently leading to cell death. Now comes the plastids. Plastids are double membrane bound organelles found in the plants and algal cells. They perform vital functions like photosynthesis, food storage, and pigment synthesis. They have their own DNA and ribosomes, allowing them to synthesize some of their own proteins. The general features of the plastids are they are present only in the plant cells. They are surrounded by double membrane. They contain their own DNA, RNA, and ribosome. They are semi-autonomous, can convert from one type to another, and they develop from proplastids in the meristematic tissues of the plants. The three main types of plastids are chloroplast, chromoplast, and leucoplast. The chromoplast are colored plastid found in plant cells other than the green plastid. They contain pigment called carotenoids, which give yellow, orange, and red color. They are commonly present in flowers, fruits, and some roots such as the carrot. The main function of the chromoplast is to impart bright colors to the different plant parts, which helps in attracting insects and animals for pollination and seed dispersal. In addition, they also help in the storage of pigment, contributing to characteristic color of fruit and flowers, especially during the fruit ripening. During the fruit ripening, the chloroplast gets converted into the chromoplast and chlorophyll disappears, while carotenoid pigment appears. For example, the green tomato converted into red tomato. Chromoplasts do not contain chlorophyll, they only contain carotenoid pigments. The chloroplasts are green double membrane bound plastids found in the plant cells that contain the pigment chlorophyll. They are the site of photosynthesis, where light energy is trapped and converted into the chemical energy in the form of food and stored in the plant for further usage. So the chloroplasts are the green and double membrane bound pigment. The chloroplasts are found in the cytoplasm of the plant cell, primarily concentrated in the mesophyll tissue of green leaves and stems, which are the site for photosynthesis, and they may also be present in other green parts and even some non-green photosynthetic tissues like the kelp embedded within the cells. The relationship between chloroplast and chromoplast is that chromoplast can develop from chloroplast through the process of interconversion. The chloroplast is bounded by double membrane envelope consisting of outer and inner membrane. Inside the chloroplast is a fluid-filled matrix called the stroma, which contains the enzymes for ribosomes and starch grain. Now comes the leucoplast. Leucoplasts are colorless plastid found mainly in the storage tissues of the plant such as the root, tubers, seeds and endosperm. They lack pigments like chlorophyll and therefore do not take part in photosynthesis. Their primary function is the storage and reserve of food. The general features of leucoplast are they are colorless plastids found in non-green parts of the plant surrounded by a double membrane derived from proplastid and can convert into other plastids. The leucoplast can convert into chloroplast when exposed to light, for example, potato tuber turning green when exposed to sunlight. Now comes the vacuoles. The vacuoles are the membrane-bound cell organelles enclosed by a single membrane called the tonoplast. They are large and prominent in the plant cells where a single central vacuole occupies most of the cell volume. And in the animal cells, vacuoles are smaller or temporary. They contain cell sap which includes water, sugar, salt, pigments and the other waste materials. This is the main difference in the vacuoles in the plant cells and the animal cells. Their main function is the storage of food, water and waste, maintenance of turgor pressure which provides rigidity to the plant cells, regulation of osmotic balance and in some cases storage of pigments and toxic substance. Thus the vacuoles play an important role in maintaining cell shape, growth and internal balance. In the animal cells, vacuoles are usually small, temporary and less prominent. Their main function include the storage of food, water, waste material, helping in transport of substance within the cell. They also assist in maintaining the osmotic balance and in some animal cells they play a role in digestion and removal of waste, thereby keeping the cell clean and functioning properly. When the amoeba surrounds food particles using the pseudopodia, the food is enclosed inside a vacuole within the cytoplasm. So each cell acquires its structure and ability to function because of organization of its membrane and organelles in a specific way. Now let's discuss the cell division. The cell division is a biological process by which a parent cell divides to form two or more daughter cells. In the cell division, actually the parent cell is dividing to form two or more daughter cells. It is essential for growth, repair and reproduction in the living organisms. The cell division ensures that genetic material DNA is accurately copied and distributed to the new cells. There are two main types of cell division. Mitosis. Mitosis is a type of cell division in which single parent cell, single eukaryotic cell divides to produce two genetically identical daughter cells, each having the same number of chromosomes as the parent cell. It is essential for growth, repair and asexual reproduction in multicellular organism. This is the different stages of the mitosis. Characteristic of mitosis is that it produces two identical daughter cells, maintains the same number of chromosomes as the parent cell. It occurs in the somatic cells of the body, that is, it takes place in the cells which are not in the reproductive cells, and it involves one nuclear division. The binary fission in bacteria is where a single cell duplicates its DNA and divides in half to create two identical bacteria. Vegetative propagation. Plants reproduce via runners, tubers or bulbs, all relying on mitotic cell division, and budding is also mitotic cell division. Mitosis in human occurs in somatic cells as discussed earlier. All body cells except the sperm and the egg for growth, repair and replacement of tissues like the skin, bone marrow and muscles, creating two genetically identical diploid daughter cells with the same number of chromosomes as the parent cell, which is essential for developing from a single zygote into the complex organism. It increases the number of cells to form the larger organism. Now comes the meiosis, the second type of cell division. Meiosis is a specialized cell division process for sexual reproduction, where one diploid cell divides twice to create four genetically unique haploid cells, each with half the original chromosome number, ensuring genetic diversity and proper chromosome count for process of fertilization. A type of cell division, it is a type of cell division where one cell divides twice to produce four genetically different daughter cells. These cells are haploid, meaning they have half the number of chromosomes, example 23 in humans of the original parent cell, and diploid means 46 in the humans, occurs in the germ cells. Now phases of meiosis, easy mnemonic to learn this is please make a T. P for prophase, M for metaphase, A for anaphase, and T for telophase. This is a table to show the difference between the mitosis and the meiosis. You can easily memorize both the cell divisions through this table. Thank you for watching. Please do like, share and subscribe.